Welcome everyone to the 74th Annual ACA Conference in Denver. I'm Alan Oliver, in case you don't know me, and I'm the current president of the ACA. I'd like to very quickly and very briefly introduce the council members. If anyone on council could please stand and just wave to the crowd. These are our representatives, your representatives on council. We have a wonderful program here that the program committee has kept on track throughout the last year. I'd like to thank in advance the program committee, session chairs, SIG officers, and all of the presenters, both posters and oral sessions, for their contributions to the program. Furthermore, I encourage everyone to introduce themselves to and help guide first-time attendees and any mentees that might have a bright pink sticker on their badge. Folks that are wearing a green sticker, and some of you will have already heard this spiel, are mentors, and we're here to help. If you're a new first-time attendee or new to the conference, please seek one of us out. We're here to help you. We can guide you. The Young Scientist SIG and VP Audette have already worked on this with the mentor-mentee program and the new members introduction that just occurred about 30 minutes ago, finished about 30 minutes ago. I'd like to remind everyone that we do have a code of conduct here at the ACA conference. You can find that on the homepage of the conference notes. I'd like to now acknowledge the peoples of this land on which we host this conference. As we gather in the vibrant city of Denver for the 74th Annual Conference of the American Crystallographic Association, we acknowledge that Denver is situated on the traditional territories of the Cheyenne, Arapaho, and Ute nations. These indigenous peoples have lived on and cared for this land for generations, fostering a deep connection to its natural beauty and resources. We recognize the historical and ongoing impact of colonization, forced removals, and systemic injustices that have disproportionately affected Native American communities. We also honour the resilience, strength, and the contributions of Indigenous peoples in the face of these challenges. As we engage in meaningful discussions, let us carry this acknowledgement forward with us. May it serve as a reminder of our collective responsibility to create a conference environment that is inclusive, respectful, and mindful of the diverse perspectives and experiences that enrich our professional community. In the spirit of unity and understanding, let us commit to fostering a conference atmosphere that honours the land, its original caretakers, and the shared journey towards a more just and equitable future for all. I'd like to now introduce Sam Powell from the Program Committee. Enjoy ACA 2024.
It's with great pleasure that I get the honor of introducing this year's keynote speaker, Professor Cathy Drennan, the John and Dorothy Wilson Professor of Biochemistry at MIT. Cathy's interest in crystallography and vitamin B12 started early when she obtained her Bachelor's of Arts in Chemistry from Vassar College. Following her bachelor's, she took a more uncommon route to becoming a professor and worked as a high school science and drama teacher for 3 years before pursuing her PhD. She earned her doctorate from the University of Michigan, studying vitamin B12 protein interactions with X-ray crystallography, furthering her passion for proteins and the structural sciences. She then went on to a postdoc at Caltech before ultimately deciding to join the faculty at MIT. Professor Drennan is an educator and researcher and has taught introductory science courses at MIT for 20 years. As part of her HHMI-sponsored education research, she co-authored a teacher training guide, What Every Teacher and Mentor Should Know, a guide to identifying and reducing stereotype threat to maximize student performance, just one of her more than 190 publications. She is also the recipient of numerous teaching and research awards. Among these are MIT's Baker Award for Excellence in Undergraduate Teaching, a Sloan Fellowship, and the Dorothy Hodgkin Award from the Protein Society. She's a fellow of many societies, including the American Academy of Arts and Sciences, and recently joined the ranks of ACA Fellows. She was also excitingly elected as a member of the National Academy of Science just last year. The focus of her career has been the study of how enzymes direct and coordinate the reactivity of metal centers to affect challenging reactions. She is well-versed in both X-ray crystallography and electron microscopies to study these interactions and transformations within enzymes, covering much of the structural sciences in her approach to understanding enzyme mechanisms. This evening, she'll present her work titled Shake, Rattle, and Roll, Capturing Snapshots of Proteins in Action. Without further ado, please help me in welcoming Professor Cathy Drennan.
Well, thank you very much. So we're all here in Denver to talk about structural science, the output of which is a coordinate file where you have an XYZ coordinate for every atom of your protein structure or other structure, if you are so lucky. So let's think about this for a minute. So in the human body, there are tiny cells. Inside those tiny cells, there are even smaller protein molecules. And yet, we are able, with the methods of structural science, to be able to obtain atomic coordinates for those atoms in those protein molecules. This is incredible. And if you are somebody who has solved a structure, you want to share that excitement of that structure. Why a molecule is so important. And this is a major theme of the meeting this time. So we have tomorrow, sharing excitement and appreciation for structural science, part 1 and part 2. We also have engaging students with structural science and visualization of science. So there's a lot of wonderful symposiums and sessions going on at this meeting. A lot of them run at the same time, so you'll have to make some hard decisions. But even if you cannot attend these sessions, I encourage you during the course of this meeting to think about how we can share excitement and appreciation for structural science, because it really is absolutely amazing. So what I thought I would do tonight is tell you about one protein, and that protein is ribonucleotide reductase. And I'm going to start tonight telling you how it is that I came to study ribonucleotide reductase, and then tell you a few things that we have learned about ribonucleotide reductase, that there's no way that we could have figured these things out if it weren't for structural science. Okay, so first, how did I come to study this enzyme? And this story starts with my late mother, Dr. Mildred Stroop-Lushinsky, who in this picture was a graduate student at Cornell University studying anthropology. And as an anthropologist, she struggled to understand what it was that I was doing when I was in graduate school. And she would say, it's so stressful when neighbors come over for dinner and they say, hey, what's your daughter Cathy doing? What is she doing? Do I need to explain what crystallography is? It was very stressful to her. So I had an idea. My idea was, save up and buy this textbook. Because in this textbook, there was a figure of the structure that I had solved as a graduate student in Martha Ludwig's lab. And I could put a bookmark in the textbook. And so when neighbors asked, she would open the book and point. This is what my daughter did. Brilliant, right? So I gave her this book, and then the next time I visited, I said, how is this working out? And she said, it was a lot of work. A lot of work? The page is bookmarked. You open, you point. How hard is that? And she said, but I finally finished. How many of you have read a biochemistry textbook from cover to cover? Let the record show no hands are going up. Okay, so my mother read the entire textbook, and she said, so I figured out what you should do when you start your own lab. Oh. I said, okay. I said, what was it, Mom? And she said, I wrote it down. My mom was one of the most organized people on the planet. She had these manila folders that were titled very carefully, all in alphabetical order. And there was one entitled, What Cathy Should Study When She Has Her Independent Career. So she pulled out the folder, she opened it up, and you know what it said, right? Ribonucleotide reductase. So I said, Mom, that's an excellent choice. Ribonucleotide reductases are really important enzymes. They're important in cancer drug targets. They make deoxyribonucleotides, which are the building blocks of DNA. Excellent choice. But I said, you know, I'm pretty sure it's such an important enzyme that there's probably every structure that you would want has probably already been solved. And so I'm not sure what I could contribute to this. Well, I could not have been more wrong about that. So I kind of forgot about ribonucleotide reductase for a while. I went on to do my postdoc with Doug Reese at Caltech. And then I was recruited to MIT by Professor Joanne Stubbe, one of the world experts in ribonucleotide reductase. So my fate was sealed. So that is how I came to study ribonucleotide reductase. So let me give you an introduction to this enzyme. And let's start with the abbreviation. Ribonucleotide reductase, abbreviated RNR for ribonucleotide reductase, not to be confused with the other kind of RNR, which is rest and relaxation. And you should not make this mistake because RNRs are extremely hardworking enzyme. And as I mentioned before, they're also major drug targets. I mentioned cancer, but it's also been proposed that if we could find differences between the human and bacterial RNRs, it could also be a good antibiotic target, and that is work in progress. So these are hardworking enzymes. What do? Again, they convert ribonucleotides to deoxyribonucleotides with this removal of this hydroxyl group in the form of water. Reducing equivalents come from a pair of cysteines in the active site that form a disulfide. So this is a hardworking enzyme because it's an important reaction, but also because most enzymes have one substrate, whereas RNR has 4 substrates. So it works on all 4 ribonucleotide substrates. And if the cell makes too many overall deoxyribonucleotides, it's toxic to the cell. If it makes too few, it's toxic to the cell. If it makes the wrong ratios, it's toxic to the cell. So RNR is regulated allosterically. And so if the cell has made too many overall deoxyribonucleotides, DATP binds, turns off the enzyme, whereas ATP can bind and turn it back on. And in terms of the 4 substrates, that can be controlled by another allosteric site. And here, DATP in lower concentrations increases the preference of the enzyme for CDP and UDP, whereas DGTP increases the preference for ADP, and DTTP increases the preference for GDP. All right, so that's pretty hardworking, but I am not done. Before you can do even one single round of turnover, there needs to be a radical species generated in the active site. So there are 3 different classes of metallocofactors that generate a cysteine or thiol radical species on every round of turnover. So how does this happen? How do you generate radical species in your active site? And the cysteine is kind of buried in this 10-stranded barrel fold, and it needs to form a radical species. How do you do this? Well, if you're the human ribonucleotide reductase, or if you're the well-studied E. coli ribonucleotide reductase, then you have a second subunit that has a stable radical, and in every round of turnover, subunits come together and the radical is transferred, i.e., really the most complicated thing you can possibly imagine is how this works. Okay, so let me introduce you to the structures now. These structures were solved before I started working in the field. They were solved in Sweden by labs of Ullin, Eklund, and Nordlund. And so we have the catalytic subunits, and so it's a dimer. This has the active site where the substrate is going to bind, and it also has the allosteric sites. So we have the specificity site that moderates the preference of the enzyme for a particular substrate, and we have the activity sites where DATP will bind, and it's this region that's in green at the N-terminus called the cone domain. So DATP binds, turns the enzyme off, ATP can restore activity. So that's the alpha-2 subunit, the catalytic subunit. And then there's the beta subunit. And so this has a stable a tyrosyl radical species that is generated by this di-iron site. And then on every round of turnover, the radical is going to go up and come back. So these two subunits have to come together, and the radical's transferred. So what do we know about the radical transfer? And so some of that is shown over here. So the radical is transferred over a very long distance between the subunits, about 35 angstroms according to spectroscopy. And in the transfer, you're transiently forming amino acid radical species. So we have, oops, let me go back. We have down here, these are the parts of beta. So there's a tryptophan. It's known to form a radical, but it's not clear if that radical is on pathway. We also have tyrosine 356. That is definitely on pathway, but it has not been visualized in any of the structures that were determined. So beta has this disordered C-terminal tail, and that's where this tyrosine 356 is. So we know something about this tail. Alpha-2 is not very soluble without beta, and so when Ullin and Eklund first crystallized alpha-2, they added a peptide with the residues of this tail, and so some of them are visualized. You can kind of see it over here and maybe over here. So we know where the very end of beta should end up, but there are 20 amino acids, including 356, that have never been visualized. So to form a radical at 356, you have to deprotonate. So there also has to be something that is a proton acceptor there. We don't know what that is. Again, this region is disordered on that tail. And then in alpha, the residues that are involved, 731, 730, and here's the cysteine that forms the radical, and there's the substrate. So the radical goes up by proton-coupled electron transfer and back down again. How on earth does this actually work? So that was one of the questions that we really wanted to answer. Okay, so one of the stories I'm going to tell you about, trying to get this structure of a complex between alpha-2 and beta-2 to understand how this long-range radical transfer occurs. And so in the early 1990s, Ullin and Eklund published this docking model where they just put beta together with alpha based on shape complementarity and the two-fold axes. And so this was really the working model for an active RNR for 30 years. And I know what you're thinking, well, what about AlphaFold? And AlphaFold gives us the same docking model. All right, so can we do better? Can we get an experimental structure to really understand how these residues line up? How do you do this amazing chemistry in class 1A ribonucleotide reductase? And the other thing I want to tell you about tonight is about the inactive structure. So we want to talk about the active structure, but also the inactive structure. And again, E. coli is our model system, so everything I'm going to show you today is from E. coli. And so we knew when we started this work that DATP and ATP bound in these green regions in the cone domains, but how does binding of something there turn on and off the enzyme? So I'm going to start with this because that's where we started, and I think it's important to do this story in order. Okay, so first question, what happens when DATP binds? So the first experiment I'm going to show you was done by a former research specialist in my lab, Ed Brignall, and Ed loves EM. And so he did some negative stain originally and saw right away that there's a difference in oligomeric state depending on whether you had DATP present or not. And so without DATP, it's mostly dissociated particles, but with DATP, form these rings. And so he was able to do then reconstruction, and he found he could place in an alpha-2, the catalytic subunit, a beta-2, the radical subunit, another alpha-2, and another beta-2. So this was an alpha-4, beta-4 ring structure. And the fact that this formed with DATP was consistent with some data also from Sweden with the GEMMA mass spec technique that said alpha-4, beta-4. But we weren't expecting it to be a ring. This is like 100 angstroms whole, and the cell is crowded. So is this a relevant structure? So this is low resolution. Let's get higher resolution and see what that looks like. So Christina Zumani, who was a graduate student in my lab at the time and is here at this meeting, so what she did was get a 3.5 angstrom crystal structure, and sure enough, it also looked like the same ring. But at 3.5 angstroms, she could look into the cone domain and see that DATP was bound. And so we could think about how DATP binding to this interface between alpha and beta would stabilize this ring. So again, we saw the ring. And I'll just mention that if you love crystallography or if you don't, there are many wonderful sessions about advances in crystallography. And I feel like we hear a lot about advances in cryo-EM, but there are a lot of advances in crystallography as well. And if someone walks up to you and says, got crystallography, you want to be able to answer yes. So I encourage you to at least attend one of those sessions. All right, but Christina's structure, we still had a 100 angstrom hole. Was this relevant? So what we did was we turned to X-ray scattering, small angle X-ray scattering, and this was work from Nozomi Ando when she was a postdoc in the lab. And I think Nozomi's not at this meeting, but I believe that she sent a bunch of people from her lab. So look for Ando on some talks and posters this week. Okay, so there are actually quite a large number of sessions about scattering techniques, and I encourage you to attend some of them because it can be a really powerful method. And one of the things I love is actually a very simple part of this, which is that you can just do a scattering profile. So you can take your protein, if you know what is a physiologically relevant concentration of your protein and of nucleotides, you can put them in solution, and you can measure the intensity of the scattering versus Q, the vector from the beam stop, and you can learn about the molecular weight and also the radius of gyration from the shape of the curve. You can plot the structure that you have, a crystal structure, an EM structure, and calculate the scattering from that and match it to solution. And this is a fantastic match. So sure enough, we have a ring with this hole. That is the structure from EM, from crystallography, from small angle X-ray scattering. And with that structural information, we could understand how allosteric regulation of activity works. So here we have the docking model again. When you add DATP, apparently it goes to this ring structure, and then ATP will shift the equilibrium back. We can understand why this would be inactive. Remember, in active R&R, you need to be able to transfer radical species over this 35 angstroms. In the ring structure, that distance is 60 angstroms, and that tail that has those residues that are essential for the radical transfer are just kind of, they're disordered, they're just floating around. So this can't possibly transfer a radical. And this was not at all what we were expecting allosteric regulation of activity to work, but it's kind of brilliant when you think about it. So like if my hand is beta and my chest is alpha, and you want to generate some deoxyribonucleotides, you can bring beta up, it can do the radical chemistry, but then when you've made enough, you put beta here. But you don't want beta to kind of, beta's, might sneak back, and you don't want that to happen, so you put it in a ring. Beta can't go anywhere, it can't wander around the cell, can't generate radicals, but it's there when you need it. I think this is a great way to do allosteric regulation of activity. And we can even get a few more snapshots about how the ring can disassemble when ATP levels rise. And so Ed Brignall went back to his negative stain. And so here is the inactive alpha 4 beta 4 ring. And again, the docking model over here. And we can see the ring kind of breaking apart. Beta's getting free, it's getting free, we're moving apart. And then beta's kind of coming in. And this is almost to a position that can be active. So when you substitute ATP for DATP, you break open the rings and you're ready to go. There is no way that we would have been able to figure out what was happening without structural science methods. In fact, people have been studying this enzyme for decades, had no idea this ring was forming. So this is an example of what you can learn from structural methods and the combination of methods. Okay, so what about an experimental structure of the active form? So why was it so hard to get a structure of active R&R? Well, I already told you one reason, which is why I did the story in this order, that E. coli class 1A R&R, again, our workhorse for studying R&R, is a mixture of oligomeric states. So that obviously complicated our structural analysis. But when we saw the ring structure, we had an idea, what if we make a mutation that at the alpha-beta interface you can't form a ring? Then can we get the structure of the active form, shift the equilibrium to alpha 2, beta 2 from alpha 4, beta 4. So we tried it, we could break the ring, but we couldn't get the active structure because of point 2, which is that the affinity of beta for alpha is modest and K-off is very, very fast. So beta is basically binding to alpha, sending the radical up, radical comes back, beta leaves. So this is why it was so hard. We needed a way to trap alpha with beta to be able to capture that structure. Okay, so we did get the structure. How did we do it? There are 4 major things that we needed to be able to finally get this structure. Number 1, money. So there's a session at this meeting from NIH. I encourage you all to go. I have been very fortunate to have NIH funding throughout my career. I am deeply grateful to NIH. But if I wrote a specific aim, said, people have been trying for 30 years to get a structure of the active state, give me money, I'll try too, I would not have been funded. But what HHMI said was, can you think of something really high risk that NIH would never fund and try that? And I said, yes, I can think of just such a thing. It is arguably impossible. I will do that thing. So becoming an HHMI investigator encouraged me to take this risk and go after the structure. Okay, number 1. Number 2, I already alluded to this, we needed a way to trap alpha and beta together. And so what I also needed is amazing collaborators like Joanne Stubby and Dan Osera, who were studying proton-coupled electron transfer in RNR for decades and putting in unnatural amino acids. And so you need a collaborator who can do years and years of background work for you. Okay, so what they did was they put in all of these artificial amino acid, nitrotyrosines, fluorotyrosines, and when you put these unnatural amino acids in this radical transfer pathway, it changes the redox potential, and it said stalls the radical, and that stalls beta from leaving. So what finally worked was a construct that was discovered by a visiting scientist from China who came to Joanne's lab to learn how to purify RNR, and Joanne said, here, purify all of these things for me while you're here. And so she found that with wild-type alpha, a trifluorinated tyrosine at 122, a single site mutant in beta, E52Q, that this together formed a competent radical transfer pathway, one product, again, alpha's a dimer, one product, and it stayed trapped for 15 minutes. So 15 minutes is nice and slow, but it's not really crystallography slow. You can't grow a data quality crystal in 15 minutes. So this brings us to the thing we needed, number 3, which was the cryo-EM revolution. I got it. Okay, so all of you in this room probably know about this revolution, the development of the direct electron detector and what that has meant in terms of the resolution that can be achieved from cryo-EM. And remember, we wanted to see side chain positions, so it was really important to us to have a certain resolution. And so I just gave you an example here of this revolution from my lab. And so this is actually the human ribonucleotide reductase, which forms an alpha-6 rings. RNR loves its rings. So this is alpha-6 ring for the human enzyme. And so with a standard CCD detector and an older scope, we got about 20 angstroms. Just putting a new detector on this scope, you go from 20 to 5.5, and then a new detector with a Titan Krios, you go to 3.3. Same exact batch of protein, 20 angstroms to 3.3 where you could see side chains. Now that is a revolution. And if you haven't joined the revolution yet and you're coming to this meeting thinking I'm not going to go to the cryo-EM sessions, I encourage you to go to some of the cryo-EM sessions, and I've highlighted one of them here. Okay, thing 4, the most important, the right student. So let me introduce you to Kenny Kang. Kenny was a graduate student in the chemistry department at MIT, and this was his project. So I'll never forget his 4th year meeting. So in chemistry, 4th year, you sit down with your advisor and there's a form to fill out. Question 1 on this form is expected graduation date. In Kenny's 4th year, we did not yet know if this project was possible. So what to put for the expected date? So I looked at the form, Kenny looked at the form, I looked at Kenny looked at me, we looked at the form again, we looked at each other, and Kenny finally said, Cathy, I've been thinking a lot about this before this meeting. And if it's okay with you, and I prepared myself, plan B, project B, okay. If it's okay with you, I would like to stay as long as it takes. I said, that seems like an excellent plan, and then realized that I should add, but we might have to reevaluate this plan in the future. But Kenny actually graduated, he has his PhD, and it was shortly after that meeting that everything started to work. And I think that R&R, which is a hardworking enzyme, needed to know that Kenny was all in. So what did Kenny do? Well, Kenny optimized and optimized, and anyone who says that cryo-EM is faster and easier than crystallography doesn't really understand the screening of grids process. So you screen for crystallization conditions, and you screen for good grid conditions. You're still screening. So screen, faster, faster. 15 minutes the particles dissociate. We didn't want to wait 15 minutes. How fast could we make these grids? So optimize, faster, faster, hold your breath. Now, literally hold your breath, because you breathe, it gets icy. Hold your breath, and Kenny got a structure at the resolution we needed to see the side chains. All right, so let me show you a little bit about this structure. So first, comparing to the docking model. Docking model is beautifully symmetric. Same with the AlphaFold model. Kenny's structure is decidedly asymmetric. So both betas are talking to a single alpha. So both betas talking to one alpha. Keep the asymmetry in mind. I'm going to come back to that. All right, what about these 20 missing amino acids? So here we have a star where the last residue of beta that was ever visualized in a structure. And here's the first residue of that little peptide that was bound to the alpha subunit. So here's alpha, here's substrate. And so we had these two positions, and 20 amino acids are missing between them, and this is 24 angstroms. So let's zoom in. Could we see density between those two positions? And the answer is yes. The density went up to the substrate, turned, and came back and connected here. So it went all the way up and all the way back. And so Kenny said, I wonder if I can put 20 amino acids in that, and he was able to. You see, goes up, comes back, just was 20. He's like, what about 19? Is 19 as good? Nope, there was extra density. What about 21? Can I squeeze in 21? Nope, doesn't fit. Exactly, those 20 amino acids have been visualized. And at that point, we're like, we better let Joanne Stubby know that we've made this progress, because she was trying to be patient in her own Joanne way. And so we thought, okay, we're ready to show her this. So it was a Thursday at 5 p.m. Kenny sent an email, Joanne, what are you doing Friday morning? I have something to show you. He sent the email, got up from his desk, went to the bathroom, walked out of the bathroom, there was Joanne Stubby. So she got the email. Her office is 2 buildings away. She got the email, ran out of her building, through another building, into our building, up to the 6th floor, ran into the lab. Where's Kenny? Someone said, I think he went to the bathroom, searched for the nearest men's bathroom, and waited outside. So Kenny came, knocked on my door, and said, Joanne's here. So we went down and we looked at this. She saw that those 20 amino acids had been visualized. We had continuous density. She leaped out of her chair, threw her arms around Kenny. In her telling, there was a kiss. I was there was no kiss. But there was one very, very happy professor who had been waiting a long time to see those residues. All right, so even before Kenny built the side chains in, this structure told us something. So it explained how beta knew that substrate was there. You don't want beta to bind before there's substrate. You don't want to send a radical up to an empty active site. That's asking for inhibition. So how does it know? Well, it goes right up to the bottom, van der Waals distance to the substrate. So it goes right up. It knows that it's there. And it also answered the question of how do you know, why does it have to leave? If it's so hard to make this radical transfer pathway, why don't you just leave it intact? Why do you have to dissociate? But we could also see that beta was filling up the active site, so product couldn't leave while beta was there. And that's shown a little bit better in this slice through. If we sort of slice through the space filling, here's the substrate, alpha's in blue, beta is in red, you can see that beta is filling up. You can't have product leave while beta is there. So beta must have to leave for product to go. So we look further into the beta tail binding pocket, and Christina Zumani, when she had been in my lab, noticed this hairpin in the alpha subunit moves when substrate is bound. So when substrate and its cognate specificity effector are bound, then you have this hairpin and it moves. And we thought, why is this hairpin moving? It's not that close to substrate. What is the hairpin doing? Well, it turns out the hairpin was forming the beta tail binding pocket. So here in orange is the beta tail that goes right up to substrate, turns, and comes back and goes out. And so only when you have substrate there do you have this movement, do you form the beta tail binding pocket. So it's orchestrated, this happening. So what about the residues of the pathway? Oh yeah, that shows that it moves. What about the residues of the pathway? So here was the sort of view before. There were a lot of question marks. And here is the structure from this trapped cryo-EM structure. And so now we can see that the distance was really pretty much on. We measure 32 angstroms here, but if you actually measure this way, it's even longer. Here's the residue that's a stable tyrosine residue. Tryptophan is right there. It's most likely on pathway. Here is 356, which was never visualized before. It's right in between the tryptophan and the residue 731 in alpha. And so we measure all the distances. Here's E52, which had to be mutated to get the structure. It is not the proton acceptor, so it seems it's a little too far away, but it's right there on the pathway. So Joanne looked at this. She was, of course, incredibly happy. But then she's like, wait, there's no polar residue to be the proton acceptor or donor from 356. Maybe it's water molecules that are important for that. Could you get a higher resolution structure so that we could see the water molecules? And this is work in progress, so unpublished, but my postdoc, Dana, who's here at this meeting, is going to talk on Thursday and tell you about this. So I'm not going to say any more now, but you will probably want to attend her talk so you can send an email to Joanne. Okay. What about this other alpha? I've been talking all this time about this alpha that's close to the beta and the radical transfer pathway. What's the deal with this alpha? So I wanted to show the reduced structure of alpha 2 in this cryo-EM map so that people in the audience would go, you did a very bad job over here in your fitting. We did. And that's because this side, we think, is the oxidized structure, not the reduced structure. We had turnover already happened in the time that we made the cryo-EM grids on this alpha. And remember I said one product was produced, so one active site turned over? We think it's this active site that turned over. All right, so what we got are two structures for the price of one. How awesome is this? And so on one side, this alpha that's not talking to beta, we can see density. The disulfide is oxidized. The structure looks like the oxidized structure that had been solved by Ullin and Eklund before. The active site is empty and the beta tail is disordered. And on the other side, we have reduced cysteines, substrate bound, and the fully ordered proton-coupled electron transfer pathway. So beta apparently talks to this alpha first and then moves. And so we can put together a mechanism for this asymmetric half-site reactivity. So here we have a little cartoon of alpha in blue and beta in red. We have our reduced cysteines and our substrate bound. The next step, the radical would fire up to this first alpha, and then we would do our chemistry. And so we would have the disulfide form, and the disulfide actually kind of moves into the active site when it forms and pushes on product. Product needs to leave. It would push on beta, and beta must flip over then to the other alpha, and then product can leave. We still have our disulfide, and then this should fire on the second side, except that this is where we've trapped it. And so beta now, when I think of beta, I think of a swing dancer. The beta has its tail. It comes over, and it dances with one alpha, and then it comes over and it dances with the other alpha. And then it needs to leave so alpha can reload with its substrates again and then do this all over. Okay, so what have we learned? Lessons from structural science. The active structure is asymmetric. We have been able to visualize all the residues that line up along the proton-coupled electron transfer pathway to really start thinking about how this incredible radical transfer works. We've learned about the beta tail binding pocket, how beta knows when it can bind. Substrate binding moves the hairpin, creates the beta tail binding pocket. We know why beta must leave on every round of turnover so that product can leave, and also you have to reduce the disulfide that's formed. In terms of the inactive structure, structural science also helped us visualize this. We know that it forms a ring, and so the inactive form holds beta at arm's length, so you can't do the chemistry. We also know that the human enzyme does not form an alpha-4 beta-4 structure, and this difference is actually exciting when one thinks about antibiotic design. So in work I didn't have time to talk about, if you have a compound that binds to the cone domains in human, it stabilizes the alpha-6 ring and can be a cancer drug, and cancer drugs do this. And whereas a compound that binds at the alpha-4 beta-4 interface, they inactivate the bacterial enzyme, have no cross-reactivity with human. So human compounds that bind to this interface don't have cross-reactivity here, and vice versa. So this is very exciting in terms of thinking about designing different molecules. And I'll just also do a final plug that there are a number of excellent sessions at this meeting that talk about structural science in view of pharma and biotech. All right, so one more lesson that we learned from all of these studies, and that is that structural methods, especially in combination with each other, provide insight into biological processes that would be impossible in any other way. And so this really is, I think, the golden age of structural science. And so welcome to the golden age of structural science and the very appropriate rebranding of the ACA to the Structural Science Society. So with that, I have an acknowledgement slide. I tried to acknowledge people as I went through, but I do want to do a little shout out. I have four current people at this meeting who have posters or talks. I want to have a shout out to people who work at the facilities. Without facilities for crystallography and cryo-EM, we could not do any of this work. I've already done a shout out to funding agencies. And finally, as you may have predicted, R&R needs the last word. So let's get this party started. And R&R does not stand for rest and relaxation, but it does stand for rock and roll. And so I will end with the dance. Thank you very much.
Thank you. My question is actually about that rock and roll video. Because the beginning of that shows one of the beta dimers hanging out in the middle of the now C-shaped structure. Is there any kind of functional significance to that arrangement? Because it looks really weird.
Yeah, so all of that was animated by Ed Brignall from his negative stain data. So he saw all these different components. And one thing that I didn't really fully mention, that when beta's in the ring, so it has its tails, which you're all familiar with now, one of the beta tails is talking to alpha set over here. The other is over here. So to become active, beta needs to let go, say over there, and swing in over here, and then be over here, and then over here. So there's a lot of movement that has to happen. And so I think that, we're seeing often on maybe it's just the way it's aligned, but the beta one beta sort of breaking away from the alpha and kind of moving over toward one set of alphas on this side, away from the alphas over here. And so it does kind of raise the question to me, do they all need to kind of disassemble, or is what's happening in the cell that one beta is kind of moving and interacting with an alpha, and maybe part of the ring could be there some of the time? I cut out for time I wanted to start my talk with some analytical ultracentrification, because I'm just like, it's an oldie but a goodie. It's a structural science method. And that really shows that under active conditions, you have a rapidly exchanging equilibrium of alpha 2 and alpha 4, beta 4. So this is like happening all the time. Question over there? You can maybe shout. Yeah.
Are there any inhibitors or molecules that will find the active site and not want to leave? Does that help trap further?
Yes. That might be how we got the higher resolution structure, which I'm not giving away Thursday morning. Be there. Yes. Yeah, so wild type structure with an inhibitor.
Hello.
Yeah.
Great talk first. Second, there's one part I didn't understand. You said that the active structure is asymmetric, right?
Yeah.
But then isn't it like it has already transferred its radical, these first alpha, beta, has already transferred its radical to the first alpha, and that's why it has formed that asymmetric. So it has been, initially it has been symmetric, right?
I think initially it was asymmetric on the other alpha side, and then it became asymmetric on this alpha side. So I think it never sits like this.
Okay.
That looks beautiful. AlphaFold loves it, but I don't think it ever does that. So it talks to one side and then the other side. And that's actually consistent with a lot of data. So when you have, gemcitabine diphosphate is a cancer drug. It's used in treatment of a lot of different cancers. And they have found that one molecule of it inhibits. They're like, but it's if you're symmetric, if you inhibit this side, you should still have this side kind of going. And so how is that true? And so it does seem like one molecule, when you have this asymmetry, that works. So I think that both sides, everything is active, but just not at the same time. So it's maybe more asymmetric, at least in terms of timing, but the docking model itself, with that beautiful symmetry, it needs to go.
Makes sense. Thank you.
Yeah.
So it was really exciting enzyme. I realized that you have so many heterogeneity, right? I was wondering that when you imaged on the first page, did you see there's so many heterogeneity on the 2D structure?
So we do see, so I guess it depends. We usually, under the conditions that we are not seeing kind of we never see alpha 2, beta 2 unless we're trapping it, for one thing. And at one point, we had tried trapping it one way, and we had someone want to quantitate kind of how much alpha 2, beta 2 we were getting with this sort of method of trapping. And the person got hold of the wrong data set, and they came and they're just like, there is no molecules of alpha 2, beta 2 in this, I'm sorry. And we're like, oops, that was the wrong one. Yeah, that should have none. So we have verified if you don't trap, you see none of the complex. So we would only see it when we trap. In terms of seeing the alpha 4, beta 4 rings, you do see some of these breakdown products of them. But one thing I'll give a shout out here that, and this was work on a different RNR, not E. coli, but of Talia Levitz in my lab on the RNR and gonorrhea. When she used the chameleon to make her grids, she saw a lot more intact rings. There was a lot less particle denaturation than using a Vitrobot. So the way that we would make the grids also could make a difference. But you do see the rings falling apart in some of them. Yeah, so I'm a big fan. If you want to talk to me about the chameleon, I'm a big fan. For transient, the interface is 500 angstroms. It's really small.
We're just wondering, what did your mom think about what you found? Did she have any predictions along the way?
So I told my mom when I started started working on ribonucleotide reductase, I was like, Mom, I'm working on the thing you told me to work on. She had completely forgotten about it. I guess that like after she had used that folder and told me what to do, it might have been recycled to have a different purpose after that. So, but I tried to encourage her that she should get all of the credit. Maybe with that we should stop. Yeah, R&R says it's time to party.